I'm Ben Housden. I'm an associate professor here at the University of Exeter. So I work in the Living Systems Institute, which is a fairly new institute in the university, and its purpose is to bring together people from lots of different disciplines of research. So we have physicists, biologists, chemists, mathematicians, etc., all in the same building. And the idea is that by co-locating those people, we can start to ask some of the most challenging fundamental biological questions, as well as more translational ones, in a way that you couldn't in a more traditional departmental structure. So my group is working on trying to identify new therapeutic targets for a whole range of human diseases. We focus mostly on cancers and neurodegenerative disorders, and our approach is to use high-throughput genetic screens in Drosophila cells and then take the lists of possible candidates that we get from that, test those in human cells. The idea being that by comparing the results between those two quite different genetic systems, we can identify the targets which are most likely to translate between all biological systems and therefore have a better chance of working in people in the clinic. We use CellTiter-Glo for all of our genetic screens. So for cancer, we're aiming to reduce the viability of cells. For neurodegeneration, we're trying to increase it and make the cells healthier. So we use CellTiter-Glo as a good readout of cell viability for both of those indications. So CellTiter-Glo is a luminescence-based assay that uses luciferase to quantitatively produce light based on how much ATP is present in a population of cells. So healthier cells tend to have more ATP and they're more metabolically active, so it's a good proxy for cell viability. So in the past, we've used low throughput methods for measuring cell viability, like cell counts with crystal violet assays or cytometry. We switched to CellTiter-Glo when we started to do things in a more high throughput way because it's much more scalable than those other methods. The main advantage of CellTiter-Glo for us is the ease with which we can use it. So it's really a case of simply adding the reagent to the cells and then incubating for a while and then reading the signal on a plate reader. So we can do that very quickly and we can automate it. We can read thousands of samples per hour. If I had to describe CellTiter-Glo in 3 words, I'd say it was easy, rapid and reliable. So far, we've identified existing drugs which have potential to be repurposed to treat those diseases, and those drugs are at various stages in the preclinical and clinical development pipelines now, so we hope to have something in the clinic within the next few years. There are 2 main things we want to do next. The first is to use similar screening approaches and apply them to different diseases to get new drugs for a wider range of indications. The second thing we want to do is develop new technologies and methods for screening so we can increase the data quality and find new ways to prioritize targets so that we know we're working on the ones with the best chance of making it to the clinic. So one of the main things we're trying to do with the technology development is increase the quality of the data that comes out of screens and get the ability to prioritize which targets to follow up on and identify those which have really the best chance of making it to clinical use. So the final implication is that we can accelerate the drug discovery process, make the whole thing cheaper and faster, and ultimately lead to faster benefit to patients.